Good morning, dear students. Welcome to the world of microbes. This is a vast area and I'll try to present it concisely to suit the requirements of UG students. This session consists of the following topics: introduction, microbes of the soil, microbes of air, microbes of water, microbes of food, conclusion, and lastly the references. A small bit of advice to you all, dear students. You can break these topics and watch one at a time. Soil microbes at one time, air and water as another session, and microbes in food lastly. I, for my convenience, included all in one session. You can split it and watch it if you find it difficult to watch it at a stretch. Living organisms such as bacteria, fungi, etc., which are too small to be seen with naked eyes but visible only under a microscope, are called as microbes. These may be single-celled or multicellular. In the previous class, you all have had a video on the diversity of microbes. So, you all know about the microbes. So the microbes are single-celled or multicellular. Humans had long used microorganisms to make bread, cheese, yogurt, wine and beer, but without knowing how these processes worked. We are also discovering that microorganisms are vital to the health of the planet by being an important part of nutrient cycling. Study of microorganisms is called microbiology. It began with the discovery of microorganisms in 1674 by this person, the first picture, Anton van Leeuwenhoek, who used his own crude microscopes to observe the microorganisms from the scum of teeth, a drop of rainwater, etc. Further, Robert Hooke, he was also a contemporary of Leeuwenhoek, he described his observations about microorganisms in his book Micrographia in 1665. Microorganisms live in every part of the biosphere, including soil, water, air, hot springs, snowy regions, acidic environments, alkaline environments, several miles deep in the ocean, several miles high in the atmosphere, inside rocks, far down within the earth's crust, and also in and on us. Microbes are very diverse and include all the bacteria, archaea, protozoa, some fungi, algae, viruses, and certain animals such as rotifers. Now, one by one, we will deal with the microbes of soil, air, water, and food. Firstly, the microbes of soil. The microbes living in soil, completing their life cycle and obtaining nourishment from the soil, are called soil microorganisms. Example: bacteria, actinomycetes, fungi, algae, protozoans, viruses, etc. First of all, the soil bacteria. These are the smallest unicellular prokaryotes and most abundant and dominant group of microorganisms in the soil and equal to half of the microbial biomass in the soil. Population is about 1 lakh to several hundred millions of them for 1 gram of soil. Per gram of soil, 10 to the power of 9 bacteria. Majority of them are heterotrophs, means they depend on other organisms for their food requirements. The soil bacteria are divided into two groups: soil indigenous or the true residents, autochthonous, soil invaders or allochthonous, those who have come from the other places. The inner region of soil aggregates have higher level of gram-negative bacteria, while the outer region contains higher level of gram-positive bacteria. This may be due to polymer formation, motility of the bacteria, surface changes, and life cycle of the bacteria involved. Bacteria do not occur freely in the soil solution, but they are closely attached to soil particles or embedded in organic matter. Even after adding the dispersing agents, bacteria are not completely dislodged from the soil particles and distributed in the suspension as individual cells. Moreover, they play a major role in organic matter decomposition, transmission of diseases, biogas production, nitrogen fixation, biotransformation means chemical alteration. Organic compounds are transformed from one form to another. Common soil bacteria are Achromobacter, Arthrobacter, Agrobacterium, Clostridium, Azospirillum, Micrococcus, Bacillus, Nitrobacter, Pseudomonas, Mycobacterium, etc. The plant bacterial interactions in the rhizosphere. Rhizosphere is the atmosphere of the root. The plant bacterial interactions in the rhizosphere are determinants of plant health and soil fertility. Free living soil bacteria beneficial to the plant growth are usually referred to as PGPRs or plant growth promoting rhizobacteria. PGPR is also termed PHPR, means plant health promoting rhizobacteria, or nodule promoting rhizobacteria, NPR. Next, coming to the soil algae. The soil algae. Algae are present in most of the soils where moisture and sunlight are available. Their number in soil usually ranges from 100 to 10,000 per gram of soil, lesser than the bacteria. They are photoautotrophic, means they can prepare their own food because they are algae, aerobic organisms, and they obtain carbon dioxide from the atmosphere, energy from the sunlight, and synthesize their own food. They are unicellular, filamentous, or colonial. Soil algae are divided into four main classes: Cyanophyta, blue-green algae, examples Nostoc anabina; Chlorophyta, green algae, example Chlorella chlorococcus; Xanthophyta, yellow-green algae, example Vacheria; Bacillariophyta, the golden brown algae, example Diatoms. Now, what are the role or functions of algae in the soil? Algae play an important role in the maintenance of soil fertility. Most of the soil algae act as cementing agent or binding agents in binding soil particles, thereby reduce or prevent soil erosion. They help in checking the loss of nitrates through leaching and drainage, especially in uncropped soils. They help in weathering or breakdown of rocks and building up soil structure. Then coming to the soil actinomycetes. Actinomycetes are prokaryotes like bacteria, and they share characters of both bacteria and fungi. They are commonly called ray fungi because of their close affinity with fungi. They are gram-positive and release antibiotic substances, enzymes, immunomodulators, etc. Immunomodulators are medication used to help normalize immune system. 70% of the actinomycetes are streptomyces. The earthy odor of newly wetted soil, odor means smell of newly wetted soil soon after rains, has been found to be a volatile growth product of actinomycetes. They are more in grassland and pasture soil than in cultivated land. Luxuriant growth is favored by neutral or alkaline pH, that is 6 to 8. In the last decades, actinomycetes became the most fruitful source for antibiotics. Important members are actinomyces, actinoplanes, micromonospora, microbiospora, nocardia, streptomyces, thermoactinomyces, etc. Soil protozoans. In moist soil, most of the members of microfauna, that is the small animals, remain in encysted form. Free-living protozoa in an encysted state are able to withstand the most adverse conditions for long periods. They are characteristically small. This enables them to survive in the smallest drops of moisture and thinnest moisture films in the soil. They are very hardy and can withstand extreme conditions of temperature. The role of soil protozoa is predatory, as these eat upon bacteria and thereby regulate their population. Example: Spiromonas, Spongomonas, Trinema, Entosiphon, Amoeba, Colpoda, Vorticella, Cercomonas, Pleurotricha, etc. All these are soil protozoans. Importance of, next coming to the soil fungi. Soil fungi are Aspergillus, Rhizopus, Mucor, Trichoderma, Penicillium, Cephalosporium, Rhizoctonia, etc. These are all growing on dead and decaying organic matter. They are the natural scavengers or recyclers. Soil viruses. The bacteriophages, mycophages, or the viruses living in the soil which eat upon the bacteria, fungi, etc. They are the soil viruses. Now these are a few pictures of the soil microbes. The first picture, top left one, is that of a bacterium azospirillum which fixes atmospheric nitrogen in the soil, and the top right one is a picture of a few algae, green algae, green and blue-green algae which live in the soil. So first one is bacterium, second one algae, third one down left is a picture of a few protozoans, diagrammatic representation of a few protozoans. Then in the middle is actinomycetes, soil actinomycetes, otherwise called as ray fungi, middle picture. Down bottom right is that of soil fungi, the fungi Aspergillus, Rhizopus, Mucor, Trichoderma, Penicillium, etc., which live in the soil. Next, coming to the microbes in air. The atmosphere is a difficult place for a microbe to survive. Desiccation or drying up is the first factor that affects aeromicrobes' survival, and humidity is the second. Temperature must be in an intermediate range, as too hot temperature can denature proteins and too cold temperatures can cause ice crystal formation. Further, radiation also possesses a potential hazard for aeromicrobes, as it can damage DNA within the cells. Many different species can be in aerosol form in the atmosphere, including viruses, bacteria, yeasts, fungi, protozoans, etc. Aeromicrobiology deals with the study of airborne microorganisms and viruses, along with important particulate matter of air, especially smokes, dust, radionuclides, and pesticides. Radionuclides are the majority of our total radiation exposure comes from naturally occurring radioactive materials, including uranium, thorium, and radon. Radon in air decays into radioactive polonium, which, if inhaled, can damage lungs and even lead to lung cancer. And pesticides, of course, those which you put to your pests, they are also allergens, or they are air pollution causes. The important forms of bacteria: spores of bacillus, fungi, Clostridium, Alternaria, penicillin, yeast spores, actinomycetes, their spores, algae, spores of bryophytes, pteridophytes, pollen grains, micro insects, and viruses. Non-spore forming bacteria: Sarcenia lutea, Micrococcus luteus, which are found in overpopulated regions. Then what do you mean by aeroallergens? In 1906, Anton van Perquet introduced the term allergy to denote any altered capacity of body to react with a foreign substance. Allergy is caused by certain biological and non-biological agents present in the atmosphere. The allergy-causing agents present in the air are aeroallergens. So, what are aeroallergens? The allergy-causing agents present in the air are called aeroallergens. Now, bacterial organisms are there, microorganisms are there, fungal ones are there, viral ones are there, which are all aeroallergens. Apart from these, again there are some more groups which come under aeroallergens. First of all, coming to bacterial microorganisms. Bacillus anthracis, one example of a gram-positive rod-shaped bacteria that utilizes spore formation to resist environmental stresses. The spore makes Bacillus anthracis a highly resilient bacteria, allowing it to survive extreme temperatures, chemical contamination, and low nutrition environments. This bacteria is associated with anthrax, which is a serious respiratory disease that affects humans. Next, fungal. Another such microorganism, one example to give, that resists environmental stress is Aspergillus fumigatus, which is a major airborne fungal pathogen. This pathogen is capable of causing many diseases in humans when the conidia are inhaled into lungs. Third one is viral. An example for a viral airborne pathogen is avian influenza virus, which is a single-stranded RNA virus that can infect a broad range of animal species as well as humans and causes avian influenza. Some of the other prominent aeroallergens are house dust, pollen grains, cosmetics, etc. This is a representative picture showing air microflora or the microbes of air. First picture is microscopic view of some bacteria, and the second picture some of the air microbes. The third one is Sarcenia lutea, the bacteria. Coming to the microbes of water. Water is the elixir of life. 3/4 of earth is covered with water, fresh water and marine. Thousands of microbes live in water. The presence of disease-causing microbes in water is unhealthy and even life-threatening. Contamination of drinking water by a type of Escherichia coli can be fatal. Many microorganisms can be found naturally in fresh water and salt water. These include bacteria, cyanobacteria, protozoa, algae, etc. Variety of microbes live in fresh water. Photosynthetic algae and bacteria that use sunlight as energy thrive in the water body near the shoreline. As the water deepens, the concentration of oxygen and light in the water body decreases. Hence, the purple and green sulfur bacteria, which can grow without oxygen, dominate here. Finally, at the bottom of the fresh water, few bacteria such as methane-producing bacteria thrives. Another type of fatal microbe found in saltwater are algae called dinoflagellates, whose rapid growth and multiplication can turn water red. Red tide depletes water of nutrients and oxygen, which can cause many fish to die. As well, humans become ill by eating contaminated fish. Sometimes blooms of the dinoflagellate genus Gymnodinium occur along the US Gulf Coast when abnormally heavy rains increase the runoff of agricultural and industrial wastes. The enormous number of these organisms in water impart a distinct red color to the water, a phenomenon known as red tide. The Red Sea is supposed to have been named for the occasional blooms of the blue-green alga Trichodesmium erythraeum. Water blooms. What are water blooms? Water bloom is a rapid increase in the population of algae in aquatic system. This occurs in freshwater as well as marine environments. Typically, one or few phytoplankton species, that is the floating plants, floating algae especially, are involved and some blooms may be recognized by discoloration of water. The water color changes resulting from high density of pigmented cells, many number of these algal cells, colored algal cells. Algal bloom concentrations may reach millions of cells per ml. In 1 ml, there will be millions of cells of this organism. Color observed are green, bright green, yellowish brown, yellowish to brown or red. Their dead organic matter becomes food for bacteria that decompose it. When the dissolved oxygen content decreases, many fish and aquatic insects die for want of oxygen. So they are suffocated and they die. Harmful algal blooms, they are called as HABs. Harmful algal bloom causes negative impact to other organisms via production of natural toxins they produce. When they are overpopulated, they produce toxins. The toxin is liberated to the water. So they produce natural toxins. They provide mechanical damage to other organisms or by other means. So in all these cases, they cause negative impact. HABs are often associated with large-scale marine mortality events. The sea population dies and have been associated with various types of shellfish poisonings. Lastly, Giardia and Cryptosporidium belonging to protozoa. These two are protozoans. They form dominant and hardy forms called cysts during their life cycle. The oocyst forms are resistant to chlorine, a popular drinking water disinfectant. Even if the water is chlorinated, these cysts of these protozoans still survive and they can pass through filters used in many water treatment plants. If ingested in drinking water or if swallowed, they can cause debilitating and prolonged diarrhea in humans. So, these are about the microbes of water. Now, this is picture. The first one is picture of fresh water and marine water microbes. The microbes in water. Different types of microbes you can see in water. All these ones which we discussed till now. The bacteria, algae, diatoms, protozoans, etc. all are present here. Now we will discuss about the microbes in food. The study of microorganisms that inhabit, create or contaminate food comes under food microbiology. Major importance is for microorganisms causing food spoilage. Good bacteria, the probiotics, are also increasingly important. Microorganisms in relation to food can have one of these three roles. Pathogenic microorganisms can cause infections or intoxications. Saprophytic microorganisms play a role in biodegradation and cause food spoilage. Cultured microorganisms like probiotic bacteria are used in food processing. The pathogenic microorganisms. Pathogenic microorganisms cause foodborne infections or intoxication, and they include bacteria, viruses, parasites, and molds. Molds are fungi. It is important to note that pathogenic bacteria and viruses usually do not cause food spoilage. They are contaminated, cannot be seen or tasted. The main factors that contribute to the occurrence of foodborne diseases are why foodborne diseases are prevalent. These are the factors. The use of raw food and ingredients from unsafe sources, inadequate cooking or heat processing, improper cooling or storing. For example, leaving cooked foods at room temperature for longer periods of time or storing foods in large containers in the fridge. Allowing several hours to pass between preparation and eating of food. Inadequate reheating. Improper hot holding, meaning below 65 degrees centigrade. Food handling by infected persons or carriers of infection. Cross-contamination from raw to cooked food. For example, by cutting vegetables for salad on a cutting board where you have cut raw meat before. This is cross-contamination. So these are the main factors that contribute to occurrence of foodborne diseases. The bacteria present in food. Campylobacter jejuni. It is a common cause of diarrhea in humans as well as some animal species. Non-typhi salmonella. This causes salmonella gastroenteritis in humans. Salmonella typhi and paratyphi. This causes typhoid fever and paratyphoid fever respectively. Staphylococcus aureus causes lesions of skin such as infected eczema, psoriasis or any other pus draining lesion. Escherichia coli causes gastroenteritis. Enterotoxigenic Escherichia coli is most common cause of traveler's diarrhea. Listeria monocytogenes. The food stored for long periods of time in the fridge. This grows upon such food. This can be fatal in immunocompromised patients where it can cause septicemia and meningitis, brain. Shigella. Symptoms of shigellosis are fever and watery diarrhea. This also manifests as a dysenteric syndrome which includes fever, abdominal cramps and frequent small volume bloody stools containing mucus. Vibrio cholerae 1 causes severe watery diarrhea which can reach up to 20 liters per day. Clostridium botulinum. This causes botulism, which is a highly potent heat labile toxin also is produced that affects the nervous system. Botulism is food poisoning, during which this bacterium produces a toxin which affects the nervous system. So these are all the bacteria in food. Then coming to the viruses. Viruses, unlike bacteria, cannot multiply in food because viruses are living only when they enter a living host. Otherwise, they are particulate matters. The main mode of transmission of a virus, therefore, is by food handlers and use of dirty utensils, vessels, which transfer the virus to food, whereupon it is ingested by humans, taken in by humans. The viruses are rotaviruses and Norwalk virus, which are major causes of gastroenteritis. And there is viral hepatitis A, outbreaks, which are mainly caused by asymptomatic carriers which handle food. Lastly, the parasites. Many parasites such as helminths have a complex life cycle involving more than one host. The major route of transmission for these parasites to humans is by the route of food. The consumption of undercooked pork or beef, or the consumption of raw salads washed in contaminated water seems to be the trend. Now the following are the parasites: Taenia solium and Taenia saginata, also called as pig and beef tapeworms. Their cysts are present in the muzzle of the animal and they are ingested and the adult worm develops in the gut. The ova or eggs may develop into larvae that may invade other tissues such as brain, forming cysticercosis and severe neurological disorders as a consequence. Next, Trichinella spiralis is found in undercooked pork. The larvae can invade tissues. Giardia lambilla. This infection can be foodborne, waterborne, or spread by interpersonal contact. It causes acute or subacute diarrhea with malabsorption, fatty stools, and abdominal pain and bloating. Entamoeba histolytica. The transmission is mainly food or waterborne. The cysts pose a major problem since they are highly resistant to chemical disinfectants including chlorination. The infection is usually asymptomatic but may appear as either a persistent mild diarrhea or a fulminant dysentery. Next, coming to the microorganisms in food production. Till now we discussed about the negative impacts of the microbes in food. Now the beneficial aspects of the microbes in food. Microorganisms in food production. Most commonly used microorganisms are yeast, bacteria, molds or fungi, or a combination of these. A good example is the process of fermentation, which results in the production of organic acids, alcohols and esters. These help to either preserve the food or generate distinctive new food products. Yeast in food. Before that, let me show you the pictures of food microbes. Representative pictures of the food microbes. The top right one is Saccharomyces cerevisiae, or the baker's yeast or brewer's yeast, used mostly in the bakeries. The down middle picture is Penicillium roquefort, which is used in the ripening of cheese. Now the microorganisms in food production we were discussing about. The yeast in food production, the leavened bread and bakery products. Saccharomyces cerevisiae ferments sugars to produce carbon dioxide, the gas that gives the porous structure of bakery products in breads. It also contributes to the flavor by formation of alcohols, aldehydes, esters, etc. So yeast is used in beer, wine, vinegar, pickles, etc. Bacteria in food production. Bacteria, the fermented milk products. Lactobacillus is the bacterium, Lactococcus, Bifidobacterium. All these bacteria are used in fermentation of milk products. A variety of foods including Indian dosa, rabri, bacteria are used. Fermentation by Leuconostoc mesenteroids, Streptococcus faecalis. All these are used. Probiotics. What are probiotics? They are nothing but live food supplements used in yogurt and other fermented milk products. It includes Lactobacillus acidophilus and Bifidobacterium bifidum. A minimum of 10 to the power of 8 bacteria per 1 ml must get to the colon alive to have a significant effect. These bacteria improve the microbial spectrum in the gut and thus contribute to the following effects. So these are the following which I tell you, the 4 points. They are the beneficial effects of the probiotics or the good bacteria in our gut. The good bacteria influence immunity and hence prevent or make diarrheal diseases milder. They decrease the risk of colon cancer. The good bacteria decrease cholesterol absorption. They produce acids that decrease the pH in the gut and thus increase the absorption of minerals such as calcium and phosphorus. So we discussed about the yeast in food production, bacteria in food production. Now coming to the fungi in food production. Cheese, Penicillin roquefort and Penicillin camembert. Note that this one produces mycotoxin at 25 degrees centigrade. Therefore, cheese production must happen at 15 degrees centigrade. So, fungi are used in the preparation of cheese and dry salami. This is a type of processed meat. This also makes the use of penicillium and Scopulariopis molds for the preparation of dry salami. Then there is soy sauce. Aspergillus species, especially Aspergillus oryzae, are involved in this production. This is also a subsequent lactic acid fermentation where lactic bacteria produce lactic acid. Sake is a type of beverage, a drink which is produced using a combination of mold, Aspergillus oryzae and yeast. It is a hot drink. Lastly, coming to the conclusion. Microorganisms are present everywhere in our environment. In soil, air, food and water, and they are also called as microbes. They are observable only through a microscope. Over 99% of microbes contribute to the quality of human life. Microbes are very diverse and include all the bacteria, archaebacteria, protozoa, some algae, some fungi, algae, viruses and certain animals such as rotifers. Microorganisms play a key role in maintaining life on earth, fixing gases and breaking down dead plant and animal matter into simpler substances that are used at the beginning of the food chain. Biotechnologists can also exploit the activities of microbes to benefit humans, such as in the production of medicines, enzymes, and food. They are also used to break down sewage and other toxic wastes into safe matter. However, the above discussed pathogens and the types of disease caused by them makes clear that they are also agents of different diseases, and these infectious agents not only spread through contaminated water and spoiled food but also through the omnipresent air, and people with weak immune system become the first victims of these diseases. Control of these air, soil and waterborne pathogens is important, and today many organizations like WHO is taking care of it. Many resistant bugs are found and controlling them is a new challenge for science. Strict adhesion to regulations and other standards set for basic hygiene maintenance and antibiotic use is a necessity. These are the references. Thank you.